Thank you very much for that introduction. And yeah, like you said, I just recently moved my lab to the National Cancer Institute. So we're still sort of in the setup phase and haven't really produced anything yet, I guess. So I'll be talking about the work that we did at UQ before I left. Okay, hang on. Oh, I can't seem to move forward. All right, hang on, I'll see if I can get the pointer here as well. All right, so I'll just tell you a little bit about nerve injury. So more than 20 million Americans suffer peripheral nerve injury. And although most of these nerve injuries are brought about by accident, actually more than 25% arise from surgery, and often these have debilitating side effects. And cancer surgery is one of these common times where nerve injury occurs, and this is because the tumor will distort the tissue and the surgeons will have really hard time finding the nerves and also avoiding the nerves. So the voltage-gated sodium channel 1.7 is located on peripheral nerves, and it's been a hot research topic in the last sort of 10 to 15 years after it was validated genetically that there's a link between NaV1.7 and neuropathic pain. So there's been a lot of research groups focusing their efforts onto finding novel 1.7 inhibitors to develop new pain therapeutics. However, that hasn't really eventuated. So we thought that we could potentially repurpose this channel or repurpose sodium channel inhibitors and see if we can improve surgical outcome through visualization of nerve using fluorescently labeled NaV1.7 inhibitors as exogenous contrast agents. So this was a collaboration run by Thomas Reiner at Memorial Sloan Kettering and Glenn King at the University of Queensland and myself. So sodium channels are large transmembrane proteins, and they are integral in initiation and propagation of neurotransmission. And there are 9 different subtypes that have found in humans, and they have different distribution and different physiology. And we tend to look for novel sodium channel inhibitors in venomous creatures because their venom is filled with disulfide-rich peptides that typically target both sodium channels, but also other ion channels and receptors. So through a bioassay-guided fractionation study, Jan in the King group pulled out this new peptide called HSPS1, and it was from the Peruvian tarantula homeoma spider. And as you can see here, it's a 28 residue peptide. It has an amidated C-terminal and this classic sort of 3 disulfide bonds, which forms an inhibitory cystine knot framework. When you go to produce these peptides, because often the components in the venom is not enough to do any sort of more intricate studies with them, you need to work out whether you want to make it synthetic or recombinantly. And because most spider peptides are amidated in the C-terminal, and typically this post-translational modification can be important for activity, recombinant production isn't really an option, and it often also leads a residue in the N-terminal from the TEV cleavage. So we went about and synthesized this synthetically. It coalutes well. You can see it coalutes well here with the native, and it was also possible to make this in high yield or reasonably high yield compared to what the recombinant one yielded. So we solved the structure of the peptide, and it has this characteristic inhibitory cystine knot 3D structure. What it doesn't really have is this hydrophobic patch, which is surrounded by a charged ring, which is really common for these spider peptides and is believed to be involved in making the peptide able to bind to the membrane. Despite it not having this surface profile, it still has this sort of the tryptophans and the lysine arginine rich sequence that these peptides tend to have. So this peptide was actually pulled out in an initial screen for novel inhibitors for NaV1.1, and that was because there was a recent paper showing that NaV1.1 was involved in mechanical pain. However, when the full sodium channel panel selectivity was done, it was found to be, like most of these other spider peptides, a NaV1.7 selective inhibitor. So like most of these inhibitory cystine not peptide, it was stable in serum for up to 24 hours. So we thought this was a good candidate to go forward with and label it fluorescently and see if we could fulfill our goal of using them as exogenous contrast agents. So we took HSP1A and we did nucleophilic substitution using a Bo-Dippy NHS ester. And this was done under basic conditions in acetonitrile water. And after 10 minutes, we had our labeled peptide. So we got about 53% yield, 83% purity. And we saw a shift here in the retention time. And that is not so surprising because of the properties of the Bo-Dippy. We had 2 lysines in the N-terminal. So there were 3 possibilities of where the Bo-Dippy could attach, but we tended to see mainly mono-labeled species. And after triptych digest, we found that it was lysine 4 that the Bo-Dippy attached to. So we then obviously wanted to make sure that the affinity hadn't changed too much following the labeling of our peptide. So we used automated patch clamping using the Q-patch platform on HEC293 T cells overexpressing 1,7. And we got a little bit of drop in potency. We had 63 nanomolar for our fluorescently labeled peptide compared to 13 nanomolar for our native peptide. But we thought it was still good enough to continue with. So the theory was that we could isolate spider peptides and synthesize them, selective spider peptides for 1,7, and we can then fluorescently label them, do a tail vein injection, and we can light up the nerves that we're interested in. So 1,7 is, like I said, is expressed in the peripheral nervous system. So we saw if we could label the human vagus nerve, and you can see here using anti-NaV1.7 antibody, we see these axons well lit up by the staining. So we then use confocal imaging to evaluate whether our peptides actually could reach the intended target. So mice were tail vein injected with the fluorescently labeled peptide, and after 30 minutes they were sacrificed. And in this case, it was sciatic nerves that was dissected out. So following a transversal section of the nerves, we can see this circular pattern, which shows the tubular structure of nerve bundles and axons. And we can see when we did a longitudinal section, we can see these linear sections. And when we did a closer sectioning, we could see the epineurium and the perineurium light up, and further sectioning, central sectioning leading down to the endoneurium and the nerve bundles, we could still see our 1,7 targeting peptide lighting up these axons. And the anti-NaV1.7 labeling of the same slices corresponds well with the histological section with the fluorescently labeled peptide. So we thought this was quite interesting. First of all, we're excited that our peptide could reach our target, but it's also been debated in the literature whether the reason why these peptides can't translate into pain therapeutics is because they can't actually cross the perineurium and reach the intended target. But we don't seem to see that problem in our studies. So there must be something else that makes the NaV1.7 inhibitors not being effective pain therapeutics. So then using mesoscopic imaging conditions, we could observe a rapid and selective accumulation of labeled HSP1A, fluorescently labeled HSP1A. So again, we did a tail vein injections, and then after 30 minutes, we exposed the right and the left sciatic nerve. And in this case, we used PBS as a control. And then we had a group of animals which we injected with the fluorescently labeled peptide. And then another group of animals where we injected the fluorescently labeled peptide in combination with 3 times concentration of the non-labeled peptide. And we can see here, like we can easily visualize the sciatic nerve when the fluorescently labeled peptide was injected on its own, but there's a 200-fold difference compared to when we inject the fluorescently labeled peptide with the non-labeled peptide. So this sort of suggests that we're reaching our target and that it's a specific target. So when examined under conditions resembling a clinical setting, a fluorescently labeled peptide also performed really well. And obviously compared to PBS, we could see here a really nice lit up right and left sciatic nerve. And when you magnify it further and use the lumen fluorescence stereoscope, we can see these longitudinal axons here lit up by our fluorescently labeled peptide. So this suggests that the lumen stereoscope and labeled HSP1A could potentially be used for intraoperative mapping of peripheral nerves in a clinical setting. So we're then interested in looking at the biodistribution of HSP1A. So taking our fluorescently labeled peptide, we observe that it really only accumulates in the sciatic nerve and really nothing in the muscle, a little bit in the kidney, but none really in the brain and the liver, which is really promising. And these peptides don't tend to cross the blood-brain barrier, so we were not so worried about them accumulating in the brain. So then we were interested in looking at the pharmacodynamics and toxicity. We obviously don't want to inject something that's going to cause toxicity during the surgery. So we did, again, IV injections of 1.5 and 1.2 mg per kg of fluorescently labeled or non-labeled HSP1A. And this exceeds the anticipated concentration in humans by about 100 times. And we then measured the oxygen saturation, heart rate, and core temperature before injection, 15 seconds, 5 minutes, and 15 minutes after the injection. And any changes in, for example, oxygen saturation and heart rate would have indicated, were sort of in hallmarks of sodium channel toxicity. So these results suggest that using HSP1A derivatives would be safe to use in an intraoperative setting, as there doesn't seem to be any toxicity following injection, so during the circulation, and also after the peptide reaches its target. So in conclusion, we have shown selective fluorescent labeling of 1,7 on sciatic nerves in both humans and mouse, ex vivo and in vivo. And we've showed in vivo imaging is possible without wanted side effects, pharmacodynamic side effects. And we also see no accumulation in the liver, muscle, or brain, which is really promising. So we believe that this could be the first generation HSP1A tracer, and it has the potential to impact surgical standards of care, reducing nerve injury during surgery. So that concludes my presentation, and I want to thank the people obviously involved in this. So like I said, it was run, the project was run by Thomas Reiner at the MSKCC and his postdoc Junior Gonzalez. And it was also in collaboration with Glenn King and his PhD student Jan at the University of Queensland. And I was helped by my PhD student Buana, Akello, and Hui. We were funded by the ARC and the NHMRC. And finally, I'd like to thank the organizers for the opportunity to present some of our work here today. And I'd like to thank the audience for listening and staying for the last talk of the conference today, and I am happy to take any questions.